Research interests include plant geography, conservation biology, rare plants of Western North America, and taxonomy of monkey flowers and Phrymaceae. Naomi holds a PhD in botany from Claremont Graduate University. In 2023, she received the Peter Raven Award from the American Society of Plant Taxonomists. In 2021, she was awarded the Center for Biological Diversity's E.O. Wilson Award for Outstanding Science and Biodiversity Conservation and the Center for Plant Conservation Star Award. Naomi serves on the board of the Southern California Botanists, treasurer for the Amargosa Conservancy, and is the chair of the Public Policy Committee of the Botanical Society of America. So I am very happy to welcome you here, specifically since we don't get very many in-person talks in Southern Nevada. So it's great to have one. And with that, I think we should try to share our screen. So can everyone see our screen?
Yes.
Anyone online can confirm?
Yeah, we got you, Marcus.
Perfect. All right. So with that, I will welcome Naomi. Thank you.
Thank you. Oh, right. Wonderful. Such a warm welcome. Thank you very much. I'm really glad that I'm able to present to you all here in person, and I'm really happy to be speaking to you all on Zoom. This presentation represents the culmination of several years' work. I've been looking at the Tacopa bird's beak probably since, oh gosh, should have thought about this earlier when exactly it was. But I started looking at it in detail around 2017. And then in 2023, we received funding, and I started working with the post-baccalaureate researcher, Erica Fontanez, who's done a lot of genetic research on this species. And so I'm presenting a lot of that to you today. I'm really excited to share new and exciting information about the Tacopa bird's beak. And so we shall get started. Okay. How do I
think just like a click or anywhere.
Okay, there we go. All right, I'll start by giving you a little bit of background on the genus Chloropyron, commonly known as salty bird's beak. This is a genus in the Orobanchaceae, so that's a wholly parasitic family. These are annual hemiparasites. It's a very small genus. There's only 4 recognized species and currently 8 minimum ranked taxa. They're primarily recognized as subspecies within those 4 species. And they're also, it's a very interesting group because they all occur in similar habitats. They're habitat specialists. So they occur in saline marshes and saline alkaline meadow habitat. There's several coastal species, but also a few inland species. 3 taxa currently recognized as occurring in Nevada. So that would be the Tacopa bird's beak, Chloropyron tecopense, Chloropyron meridimum subspecies canescens, and Chloropyron meridimum subspecies perii, which was newly resurrected and then transferred into the meridimum complex. And that's a taxon that occurs in eastern Nevada and western Utah, for which there's not a lot of information on right now. It's a species I actually hope to, or a taxon I hope to explore a little bit more this summer. With my student Erica, who, she has been adventuring with me on Tacopa bird's beak, and now she's actually doing work to explore further the whole genus Chloropyron. Okay, just to give you a little background on the taxonomy of Chloropyron. So it's in the subtribe Castellani, and previously it was treated in the genus Cordylanthus in subgenus Hemistegia. So many of you may know it as Cordylanthus tecopensis. But it was segregated into, and they resurrected a genus that was originally described by someone named Baer, because they did a molecular phylogenetic work, and they basically did a phylogenetic classification of this subtribe. So this is work by Dave Tank, Mark Egger, and Olmsted, where they basically rearranged this subtribe. And what you can see here is it used to be placed, the old taxonomy placed Chloropyron in this genus Cordylanthus, but with the new phylogenetic information, it's been segregated into Chloropyron because it's in this clade here called Pseudocordylanthus, which includes 2 genera, Dichronostegia and Chloropyron, which was resurrected. So it's a very unique group and very interesting to study. Okay, just to give you a little bit of understanding of the morphology we see in this group, these are some of the members of the Castellani. This is a figure from that paper from Tank et al. On the phylogenetic classification of the tribe. And you can see they're really beautiful plants, and they display a range of floral morphologies. And so this is Chloropyron meridimum canescens here pictured. And you can see here's Cordylanthus, and you can see just sort of based on morphology, you can see why they were kind of placed together. They look kind of superficially similar. And this is Dichronostegia, which also looks morphologically similar. So these were all kind of placed together previously and now segregated into different genera. Now on to our star of the show, Chloropyron tecopense, the Tecopa birdsbeak. This is a species that occurs inland in these alkali wetlands. It's endemic to the Mojave and Great Basin deserts, and it occurs along the California-Nevada border. So it spans both California and Nevada and is known from 4 population areas, which I'll talk about in depth a little bit later. This is a rare plant. So it has its conservation status. It's ranked by NatureServe as a G2, which means it's imperiled because it has a very narrow range. In California, we use this California Rare Plant Rank system. And in that system, it's ranked as a 1B.2. And what that means is it's rare in California and moderately threatened. The 0.2 means that it's moderately threatened. The subnational ranks in NatureServe in California, it's ranked in S1, in Nevada and S2. So both narrow ranges in both states. And so in addition to having narrow range, it's subject to many threats. The threats include groundwater extraction, lithium and clay mining, geothermal energy, exploration and development, feral horses and cattle, trampling, OHV trespass, recreation. So it's sort of like a whole gamut of threats that we're seeing, sort of really an onslaught of threats we're seeing in the desert affecting this plant. You can see a picture here of mining exploration taking place at the Fish Lake Valley salt marsh, right along the edge of the playa. This exploration took place literally just feet away from plants, like mere couple of feet. So these plants are highly threatened by this exploration activity. And as a result of these threats, the Center for Biological Diversity have prepared and submitted a petition to list this species under the Endangered Species Act in 2023. That petition has a positive 90-day finding, and we're currently awaiting the 12-month finding on if this species should be listed under the Endangered Species Act. So we embarked to participate in some research to learn more about this species. I was really interested. I'll show you a map about its distribution, but basically it has very disjunct and isolated populations, and I was really curious about it and if there might be corresponding differences in morphology, ecology, and genetics. So we wanted to compare, there are currently 3 extant populations. There's one population we think is extirpated. So we wanted to compare those. And we wanted to collect more data on the distribution and abundance of the species and the threats that are active to it. And to collect that data, we used a lot of remote sensing data using Sentinel-2 shortwave infrared imagery. This is a little seedling of Chloropyron tecopense. They're very sweet. They stay in the seedling phase for a really long time. There might not even be seedlings yet right now. They probably haven't even emerged yet. But let's say they emerge like in late May and June, and they'll stay that way for like months until they finally decide to flower. And they really like it hot. Okay, so here's the distribution of Chloropyron tecopense. So way far in the north here, that's not far north for me. It gets like way too cold if I go much further north than that. But this is in Esmeralda County, Fish Lake Valley area. There are these occurrences here. And then about 150 miles to the south, we have Ash Meadows region, and here they span between Nevada and California. And then just 30 miles south of Ash Meadows, we see more populations here in Shoshone and Tecopa. This is where I first got to know the species, down in around Shoshone and Ash Meadows area. And then just 25 miles south of there is Saratoga Spring, which is in Death Valley National Park. And that's where we have done a lot of surveys. We've surveyed there like about 4 times, and we haven't relocated them, and they might be extirpated from that location. Even though that location sees a fair amount of land protection currently, there could have been changes in hydrology there. Oh, can I? Oh, hi, right there. Oh, thank you. So these are groundwater dependent species. So these are wetland plants, wetlands in the desert. Where does the water come from? So there's a huge supply of groundwater coming from a vast carbonate aquifer that expresses at springs and seeps, and upwelling of shallow groundwater. This is part of what really started to intrigue me about these rare plants of the alkali wetlands, is you have these habitats that are perennially wet in the hottest, driest place in North America, and they're supplied by this very ancient water. It's water that came down during the last ice age as snow and rain in the last ice age. You had giant lakes, you had snow-capped mountains. All that snow melted, it fed these deep carbonate aquifers. It takes a long time for that water to cycle through the system. You know, there's these deep fissures, you know, Devil's Hole, they don't even know how deep it goes, right? You have earthquakes in Japan, the hole is shaking over here in Nevada. That's like really crazy. So there's just very complex hydrology. And then at these springs and seeps, you have this ancient water that could be, you know, 5,000, 10,000 years old coming to the surface. And that water is highly threatened because it's being pumped at an unsustainable rate. And as you have increased pumping and not as much recharge, you know, you're drawing against a balance that's not like, right, like coming back up. So that's what makes these plants so highly threatened, but yet you have these incredible ecosystems like Ash Meadows National Wildlife Refuge, right? Such a global treasure. It's recognized internationally. It has the highest concentration of endemic species in North America, and just right, all the little pupfish. There's just as many endemic plants as there are pupfish, if not more. So it's just really incredible. And the bird's beak, for instance, in this habitat, this is Carson Slough on BLM land in Nevada. You can see Eagle Mountain here in the background. This is the Resting Spring Rains. This is Shadow Mountain. The bird's beak will occur kind of in these salt flats kind of areas, not necessarily in the verdant green, but more in like alkali flats. And so we use this Sentinel-2 shortwave imagery to like be able to visualize the groundwater that might not be apparent from just regular land satellite imagery. And so this is what the Sentinel-2 shortwave imagery looks like. So this is what it might look like on the ground, right? You have expression of groundwater, basically shallow groundwater that's upwelling. You have the soil is like very clay-like, and it's saturated all year round. And you have heat, drawing the water up through evaporation, through capillary action. You just have constant draw on the aquifer. And so it sustains these wetlands, keeps them wet. On regular land satellite imagery, this habitat here might just look kind of like white salt crust. And it's hard to know how much moisture is there or how that corresponds to plant distributions. But through mapping the plants and then visualizing it using the Sentinel-2 shortwave imagery, we were able to see that these points actually very closely correspond to where plants occur. And so we can look for these signatures on the shortwave infrared imagery and look for places where rare plants aren't mapped, survey those, and we have in fact documented many new rare plant locations using this remote sensing technique. We've done it for the Tecopa bird's beak, for Ash Meadows lady's tresses, and for the Amargosa wort, which are all groundwater dependent species. It's especially fun to do it looking for the ash meadows ladies tresses, because it's definitely like they occur in small populations and it's like a needle in a haystack situation, and it's very, very fun. Okay, so we have these three populations of Tacopa birdsbeak, from Fish Lake Valley, Ash Meadows, and Shoshone and Tacopa. So I'm kind of going to break down some of the information we found on these three separate populations. And I just kind of want to let you know that I'll be using these abbreviations throughout my presentation and as color coding to try to kind of stay consistent throughout so we can follow along. I'll also be denoting these species in this order, which is, it's not alphabetical order. I should have done alphabetical order. I don't know. In my brain, I'm thinking like based on geography. So it's from north to south, with Fish Lake Valley being further north and Shoshone and Tacopa being further south. So I'll just kind of always talk about Fish Lake Valley first and then Shoshone and Tacopa. Okay, so just some basic population metrics and what we found. We did a lot of surveys. We've done population estimates and just learned a lot about these plants. So in Fish Lake Valley, Nevada, we estimate upwards of 200,000 plants in that region. So it's the most abundant of the three areas, yet it's actually the most highly threatened. So there we have numerous threats: groundwater pumping, hydrological alteration, geothermal, lithium exploration, OHV, cattle grazing, and probably some other threats I don't even know about yet. They're highly threatened because there's no land protection there. All these plants, they occur entirely on land managed by the Bureau of Land Management, but there's no area of critical environmental concern designated, no wilderness, no refuge, nothing to protect them. But in southern Nevada and California, we have the plants at Ash Meadows region. They occur on land managed by U.S. Fish and Wildlife Service and BLM Nevada and California. We've estimated there about 10,000 plants. I think it might be a little bit of an underestimate. There's many unmapped populations in this region, and it could be that there's more occurrences there. There are many threats still to these populations, even though they are afforded a lot of land protection, because they do occur largely in Ash Meadows National Wildlife Refuge, the Ash Meadows area of critical environmental concern, and the Amargosa North area of critical environmental concern in California. But there's groundwater pumping and unsustainable groundwater pumping in the region, local hydrological alteration. There had been some past lithium exploration, and that certainly can arise again in the near future. Clay surface mining, feral horses, OHV, and a lot of emerging issues with solar in the Amargosa Valley, which I didn't list as threats, but there's a lot of emerging issues around that. In Shoshone and Tacopa are the smallest population area, only about 6,000 plants. And we see a lot of the same threats, but including threats by recreation, because there's a lot of hot spring recreation in that region. And so there's trampling of plants and sort of alteration of hydrology related to that recreation. But we have the Amargosa Wild and Scenic River designated and the Tacopa Ecological Reserve, which is land managed by California Department of Fish and Wildlife. And then I already mentioned Saratoga Spring, which is in Death Valley National Park. In 1954, it was noted as being locally common. It was collected there by a fellow named John Roos, who was a resident in Southern California, and he did a lot of hot spring recreation in Tacopa Hot Springs and really enjoyed learning about the plants there, and he made a lot of collections and he actually described the Tacopa birdsbeak. And it's the only collection for that particular location, and it's not been relocated. There's been a lot of different activities occurring at Saratoga prior to the designation of the park. So it's really uncertain as to what happened to the plants and why they're no longer there, and if there could have been some kind of hydrological alteration in the intervening years. I did some calculations to kind of understand a little bit more about the range of the plants in this area. So I calculated extent of occurrence and area of occupancy. And this is just sort of a take home. So the total range, if you include all three population areas, the area of occupancy is about 16 kilometers squared. And then Fish Lake Valley has the largest area of occupancy, and Shoshone and Tecopa has the smallest area of occupancy, and also the smallest range. So that's just, I'll kind of come back to that pattern a little later. Okay, so just to give you a sense of the habitats that support these plants, this is 2 different kinds of habitats in Fish Lake Valley. So around the Fish Lake Valley salt marsh, we oftentimes see very open habitat in clay soils and alkali crusted open areas near saltgrass. But then at Gap Spring, we can also see them in pretty densely vegetated areas with sedges and juncus, and this is also growing sympatrically with the other Chloropyron meridimum canescens. That's the dark stuff right here, and this is Chloropyron tecopense. We did some soil samples here, and so these soils and habitats are really amazing. They have really high pH. 8.4, very alkaline soil. They're composed, kind of mostly of sand and silt, but they do have pretty high clay content as well. And then the salinity is really kind of off the charts, just very saline. So I'm going to kind of just, I'll compare these numbers because we also did these measurements at Shoshone and Tecopa, and the numbers are quite impressive there. So here's some habitats at Ash Meadows. There are very similar. We have open habitats here, like at Collins Ranch, and sometimes the plants occur maybe at the edges of these more dense areas, but they tend to occur a little bit in more open habitats in Ash Meadows. We don't have data on soils for Ash Meadows, but I would like to gather a soil sample in that area to see how it compares with Fish Lake Valley and Shoshone and Tecopa. Because when we look at Shoshone and Tecopa, vegetation-wise, again, it's pretty similar throughout the range, vegetation-wise. But we do have a higher pH. At Fish Lake Valley, it was 8, and here we're at 9.6. Similar composition in terms of sand, silt, and clay, but the sodium, chlorine content, and magnesium too, like really high. So it's just very highly salty mineralized soils, just quite extreme environments for plants. And these plants have to be very highly specialized to live in these environments. And then we look at the climate and compare the climate between these different places. We see, of course, in the north, Fish Lake Valley has higher annual precipitation. This is the bars of precipitation, and then the lowest temperatures. And then as you get further south, you get decreasing precipitation and increasing temperatures, which we would expect from that latitudinal gradient. So these plants between at least Fish Lake, Ash Meadows and Shoshone and Tecopa have similar sort of climate conditions, but Fish Lake Valley definitely is a cooler, milder climate in comparison. And so we looked at the phenology of these plants as well, and we used iNaturalist to score phenology. I used the annotation function. I don't know if any of you all have explored the annotation function, but it's really, really helpful. And we also incorporated a physical review of herbarium specimens to confirm, and we confirmed, got additional phenology information that way. And what we see is that in terms of flowering observations, we have sort of a truncated time period. They all start around a similar time, but the plants of Fish Lake Valley sort of really abruptly end, around the September, whereas the plants in Ash Meadows and Shoshone of Tecopa, they keep blooming almost into December. And that's really probably attributable to temperature, that it just gets too cold for those plants to stick around. Remember, these are annual plants and they just die back to the ground. So these plants in Fish Lake Valley just have a very compressed growing season. And then similarly for fruits, there's a lot of overlap between fruiting and flowering because the plants are simultaneously flowering and fruiting throughout the season. And also they kind of pretty abruptly truncate around the end of September for Fish Lake Valley, but continue on in Shoshone and Tacoma and Ash Meadows. I have more data to add to the Shoshone and Tacoma. I only have 45 included in this graph, but I realized after the fact I have a ton of herbarium specimens that were unmounted that didn't get included into this data that I will add. But I don't think it will change the picture much in terms of, because a lot of those correspond to some of my INAT observations. Well, just looking at the data, how it looks sort of raw on iNaturalist, this is not broken up by the 3 different areas. This is all of them combined. This just shows that there's a lot of overlap between these phenological conditions where you have flowers and fruits kind of corresponding together here. Red is flowers and blue is fruits. And then you also have buds kind of overlapping with flowering and fruiting. But what you can see is that plants are vegetative. They start to be detectable in March, and they really don't start to flower until mid-June. So just in terms of surveys for this species, to be able to detect them and with flowers, like you really kind of need to wait till it's hot and before they even start to flower. And then this is just, I scored leaves where they have green leaves, yellowing leaves, or dead leaves. And so you can see the skeletons are apparent early in the season where you have a lot of no dead leaves for the prior year. And then they start kind of greening up, in April. And then this is around the time you get senescence in November, December. And that's really probably the signature mostly from the Ash Meadows and Shoshone plants. And just on floral visitors, we only have a little bit of data. This is an observation I made with a colleague, Chris McCready. We were out surveying and we saw this little metallic sweat bee visiting flowers. I have some genetic data to kind of further evaluate or think about breeding systems, which we think it's actually highly self-pollinating. We do find very frequently thrips inside the flowers, but haven't really seen a lot of pollinators or floral visitors very infrequently on the plants. But also I haven't done very intensive pollinator surveys yet. So I'd like to learn more about pollination in these plants. And so we also looked at morphology to kind of look to see if there were differences between these 3 areas. So we looked at plant height, plant width, corolla length, corolla width, calyx length, calyx width, and bracts, bract length and width. So we measured all these traits using herbarium specimens. And what we found, I'll break this down. We have some graphs coming up, but don't fret. We'll get through it. So just to note, I measured this for 13 plants per area, and that doesn't seem like a huge number, but it was actually really difficult to get all the same parts off the specimens to contribute to the data set, like where you can get, like when they're smushed as herbarium specimens, and to be able to get the measurement accurately. And so I tried to make the measurements so it was like we were able to get a lot more for Fish Lake Valley, but I didn't want to like skew the data towards one population area. So this is an analysis of variance, and what this and a Tukey test. And what this tells us is that basically Fish Lake Valley plants are shorter relative to Ash Meadows and Shoshone and Tacoma. So this is height. So you can see that there's a lot of individuals here on the short end, whereas Ash Meadows and Shoshone and Tacoma kind of overlap quite a bit. And that this is significant according to the Tukey test here, where it's identified Fish Lake Valley as being significantly different from Ash Meadows and Shoshone and Tacoma. Similar with plant width down here. This was also significant, where Fish Lake Valley was significantly, the width was less than Ash Meadows and Shoshone and Tacoma. And just also a lot tighter in terms of like the distribution of the measurements. And so basically the plants are smaller in Fish Lake Valley, but ash meadows and Shoshone and Tecopa were similar in size. So not really morphologically readily distinguishable. But Fish Lake Valley is like, you could once you see them a lot, you're like, yeah, these are like little short plants. And that could be related to, right, the shortened growing period and the colder weather and all these sorts of things. So we also looked at corolla length and width. And the plants in Fish Lake Valley actually have longer corollas. So they're shorter, smaller plants, but they have longer flowers compared to ash meadows and Shoshone and Tecopa. And that's also something you can see, that they're like kind of little plants with pretty big flowers. But the width was not significantly different. So Fish Lake Valley was only significant with respect to corolla length, but wasn't significantly different with respect to corolla width. And so there was no significant difference in corolla width. And we see, the same thing with bracts. I didn't present the bracts because we see the exact same thing. Okay, so then we collected a bunch of genetic data. We extracted DNA, we did dd-rad-seq, and have a lot of SNP data to analyze. And we built a family tree of the Chloropyron tecopense group and its closest relatives, all the other species in Chloropyron. And here are some, you don't have to read all the little names, but this is some of the patterns that we find. One is that Chloropyron tecopense resolves as a lineage. So they are each other's closest relative, all the Chloropyron tecopense, and that had 100% bootstrap support, so high statistical support. These are the other Chloropyron species, and the closest relative to tecopense was Chloropyron meridimum species complex. It resolved as sister to Chloropyron tecopense. This is where things got really interesting, because I had expected, in my hypothesis, I thought ash meadows and Shoshone and Tecopa would be each other's closest relatives, because they are in proximity very close to one another. They're only 30 miles apart. They're morphologically indistinguishable. They should be each other's closest relatives. Well, we have Shoshone and Tecopa resolves as a clade, like all the samples from there are a clade with 100% bootstrap support, but ash meadows is not its closest relative. Ash meadows is more closely related to Fish Lake Valley. So we have a clade of ash meadows plus Fish Lake Valley with high statistical support. And then a clade containing all of ash meadows, that's the blue clade, and a clade containing all of Fish Lake Valley, the pink clade. So basically ash meadows and Fish Lake Valley, they're reciprocally monophyletic. They each geographic region are kind of their own little lineage, and each one with high statistical support. Now, I found this very curious and interesting because I was like, why would Fish Lake Valley, which was 150 miles north, be more closely related to ash meadows than ash meadows is to Shoshone and Tecopa? And that's a question I'm still exploring. I have some ideas, some thoughts, perhaps. It could be that Shoshone and Tecopa at some point diverged, and then maybe you could have had a long distance dispersal event to Fish Lake Valley from ash meadows, and you have founder effect, and then they're closely related to Fish Lake Valley. Anyway, that's an idea. But anyway, I found that very interesting. I also found it very interesting that there wasn't any mixing between geographic regions. Each geographic region resolved as its own lineage with very high statistical support. So then we further looked into this, exploring other data. We collected data at the population level as well. And so here are some population genetic statistics, and we'll just break it down one by one. All right. So we have 183 samples in this data set, and we looked at Tajima's D. And what this, so basically when this statistic is greater than 0, it tells us that we have potentially a sudden population contraction, or maybe balancing selection is happening. So rare alleles are scarce. And so that's what that tells us. But so mean FIS was really interesting because this is the inbreeding coefficient. And the inbreeding coefficient measures the reduction in heterozygosity within a subpopulation compared to Hardy-Weinberg expectations. But what this tells us is there's an excess of homozygotes due to inbreeding. And so that is when it's a positive FIS. So that's what we have. Basically, there's a strong signature that these plants have a high incidence of inbreeding happening, which maybe is like we don't see a lot of pollinator activity, and maybe that's why. And this is further emphasized or further like supported by the fact that our observed heterozygosity is less than what was expected. And specifically for Fish Lake Valley and Ash Meadows, it was significantly less. So that's to say, yeah, there was certain that basically it suggests inbreeding and a deficiency of heterozygotes. So there's lots of indication for inbreeding happening in these populations. And then we also have this measurement of pi, which is nucleotide diversity. So this basically tells us that Ash Meadows is the most genetically diverse set of populations compared to Fish Lake Valley and Shoshone and Tecopa. And this is actually something we've seen in a lot of other population genetic data we've been gathering, both for Spiranthes infernalis and Zeltnera nemophila, the spring lemming centauri, where Ash Meadows has the greatest genetic diversity compared to any other population. And that kind of makes sense because Ash Meadows is like this, right? It has such great habitat, incredible wetland, large populations. And so it tends to support populations with higher genetic diversity. Ash Meadows also had the population with the highest private alleles. So these are alleles that aren't shared between other populations. And so there's unique genetic diversity present at Ash Meadows that's not located in the other two populations. Similarly, Shoshone and Tecopa has 10, which is not shared. So that's interesting. Fish Lake Valley only had 3 private alleles, and perhaps that related to, if it's very closely related to Ash Meadows and it's descendant from Ash Meadows, maybe that's why. So we also did some different analyses to just see how these different populations sort of cluster out. And we did a principal components analysis, and you can see there's not a lot of overlap between the populations. So we have here Ash Meadows, Fish Lake Valley, Shoshone and Tecopa. It's interesting that there's so much spread within the groups, but there's no overlap between the groups. And we further analyzed, so basically the discriminant analysis of principal components takes the first principal component and then does a further discriminant analysis on it. And then also here identified the 3 as 3 separate groups. So this is just like compounding evidence that there's not a lot of shared genetic variation between these 3 population areas. So there's just not a lot of genetic exchange at all. They're completely genetically isolated. And we see in the structure plot as well. So the way the structure plot works is each one of these lines represents an individual, and it assigns an individual to different genetic clusters. So we have these 3 genetic clusters. And you can see that basically Fish Lake Valley, the overwhelming genetic cluster is this 1. In Ash Meadows, the overwhelming genetic cluster is 2. And in Shoshone and Tecopa, by far the overwhelming genetic cluster assignment is to 3. And this was the optimal the optimal set of populations was the structure now identified that there were 3 optimal populations, that they should be segregated or identified as 3 populations. And then we also looked at genetic differentiation between them. So if you can't tell, we wanted to throw like the kitchen sink at it and like do every possible kind of analysis to kind of understand how they're related to one another. But basically what this tells us is that, so this compares Shoshone and Tecopa with Fish Lake Valley, this square does. And so basically these numbers go from 0 to 1. 1 being high genetic differentiation, and 0 being no genetic differentiation. These numbers are approaching 1. So what that says is that Shoshone and Tecopa and Fish Lake Valley are significantly differentiated from one another. The numbers approaching 1, it means like these are very, very different. These are not the same thing. Same thing between Shoshone and Tecopa and Ash Meadows, a very high number, approaching 1. And Ash Meadows and Fish Lake Valley also are significantly differentiated from one another. But the highest one is between Shoshone and Fish Lake Valley. Like these are very, very different from one another, especially for being recognized as the same species. Okay, so just to kind of summarize all that genetic information, we have in this species, the Tecopa bird's beak, low genetic variation, high rates of inbreeding, very strong genetic structure between different locations. Essentially, we have geographic isolation, genetic isolation. A lot of this might be due to founder effect and genetic drift from subsequent isolation. I would say these different populations are functionally, reproductively isolated from one another. Okay? So a lot of isolation. So it leads me to think about, what is a species? These are all treated as a single species, right? So when we think about species, there are many properties used to define species and what species are. We can look at morphology. We can look at reproductive isolation. We can look at the family tree and phylogenetics and if they form an independent lineage or not. There's really no universal set of standards in plants that are applied to all species or across the kingdom of life, right? And so, you know, there's this concept we think of called cryptic species. So cryptic species is this idea where genetic divergence outpaces morphological divergence, where you might have high genetic differentiation amongst populations that you readily can't identify them from one another. You couldn't just say, looking at them, this is A, this is B, this is C. You'd be like, these are all the same thing. At least to our eye, we might identify them in that way. And this is one classic example of this is in giraffes. So giraffes traditionally were all recognized just as one species. And then there was genetic evidence that came out that identified that there were 4 discrete lineages of giraffes. And they initially didn't have any morphological data to support that there could be 4 species of giraffe. But since then, they've done a lot of research, and they actually have found cranial data that shows that there's like dimorphism between males and females and different levels of dimorphism between males and females between the 4 sets of populations. So there's actually is some morphological divergence between them, but it was just sort of obscured or traits we weren't looking at or what we didn't see or understand. And what genetic diversity allows us to do, it allows us to visualize diversity in a whole different way, to see essentially the unseen, what we can't see with our naked eye. And we can recognize new patterns. We can see how populations are relating or not relating to each other, if they're not exchanging genes. And this is really important for conservation because we have these threats on the landscape and we designate protections based on what species are recognized and how vulnerable they are. And what if we can't actually see all the species with our naked eye? Then how do we know we're protecting the appropriate units of diversity? So I think being able to deploy these genetic studies is really important for helping us to bring to light the diversity that really exists in the landscape. And so what we're proposing is 3 taxa in Chloropyron tencopense. We're going to break it up and we're going to recognize Fish Lake Valley, Ash Meadows, and Shoshone and Tecopa as different taxa. And so the question arises, at what rank do you recognize them? Are they full species? Are they subspecies? What do we do? So I have some thoughts on it. And I'm proposing to recognize these as 3 different subspecies. And the reason I'm doing that is there's several reasons. One is through the phylogenetic data, we know that all of Chloropyron tecopensii, they all share a common ancestor. They are a single lineage. They are difficult to identify morphologically. And so I think just based on our own biases, it might be hard for other folks to swallow them as 3 different species. And then also for tradition. For tradition, because in Chloropyron, we often recognize more subtle units of diversity as subspecies rather than as full species. We only have 4 full species in Chloropyron, so it kind of would match up with how other units of diversity are recognized in the genus. And so this recognizes our own human biases, where we really clearly, strongly prefer morphology above all else, right, for diagnosing species. Here we also have geography. So if you were writing a key and you say you couldn't, how could you tell them apart? Well, you could always just say, where did they come from, right? So you can key these out. And you might say that's cheating, but really, these clearly are very highly differentiated populations, and they warrant their own recognition. Otherwise, we could have, so for instance, if all of Fish Lake Valley were lost, that genetic diversity isn't replicated elsewhere. We would be losing something forever, right? So I think it's important to recognize them as independent units. This meme was just, I don't really think taxonomy is arbitrary, but, just got to make it a little spicy. Okay, so to summarize, we are taking a total evidence approach to recognizing 3 distinct taxa in Chloropyron tecopensii. We're using geography, we're using morphology, we're using phylogeny, phenology, like population genetic data, the whole shebang. We throw the kitchen sink at it. There's probably more data we could collect. We collected micromorphological data as well using the SEM, scanning electron microscope, but we haven't really fully analyzed that, so I'm not including any of that here. These taxa may be cryptic to botanists due to the limited morphological differentiation, but I find that it's important to recognize them for conservation value and also just to recognize diversity in the world that is statistically supported, and if someone could repeat the study and find the same thing. Fish Lake Valley population has the largest area of occupancy, the largest population size, but it is the most threatened. So that's, I think, very important. Ash Meadows is the most genetically diverse, which we see repeated in other species. Shoshone and Tecopa has the least genetic diversity, the smallest population size, and the least area of occupancy. It does have a fair amount of land protection, although its water is still highly threatened. So I would say Shoshone and Tecopa is also very much a conservation concern, especially in a changing climate. I would say these species, perhaps Fish Lake Valley has a larger population. Maybe you have population expansion due to just having milder conditions, and they like it a little bit cooler and they'd rather march north, but they have very little opportunity to march north because you have these isolated habitats and a lot of area between here and there to be able to disperse between, right? But all 3 taxa are highly vulnerable to extinction and very important to recognize. So our next steps is to finalize the report and then to publish that in the scientific literature, including the description of 2 new taxa. And we're going to continue looking for those Saratoga spring plants because I don't want to give up yet. And I really want to know how they fall out in all this genetic data. Are they different from Shoshone and Tecopa? They're only 15 miles south. I don't know. According to the data, we would predict that they would be very different, right? And if they are different and we just aren't able to include them in the analysis, are they a species that are subspecies that has gone extinct? Does that represent total extinction? So with that, I thank you. Okay. How do the questions work?
I have a question. You kind of got to this about the micromorphology thing, but like in a lot of cryptic complexes like Mancelias, you need seed characteristics to identify between like species. So have you looked at the seeds?
We need to get better data on seeds. There are some reports in literature that there could be differences in seed color and seed, like texture, micromorphology. And so we want to get better data of the seeds specifically on the SEM, and then just to get better pictures with a color scale to see if there is indeed these different color differences. And so that's something we're still looking into, and that could be a character, that a morphological character that could be used. Yeah.
I believe we have some questions here. Actually, I have a question. Naomi, have you used your observation to look at the water in below Saratoga Springs to see if that might have changed since 1954?
There certainly is hydrological data being collected at Saratoga. I think that USGS and others have monitored the groundwater there. There have been just changes into the flow, how the water flows at the site. There's like a big pond and some like berming, and there also had been tamarisk removal there, and just like earth moving and things like that. That could have the hydrology is so important to these species. When you redirect the flow or dry up the habitat, if the population really only existed in a particular spot, just drying up that habitat, right, could cause these species to go extinct. Like that's just demonstrates, I think, how important it is to maintaining hydrological function at the sites, because you could have extinction.
Thank you. We have another question. John's just going to come up. He has to come up to the screen to ask.
So these plants are hemiparasitic. Do we have any information on what the host plants are?
Yes. I forgot to mention that. Their host plants are generally thought to be grasses, so Disticus and Sporobolus. Many taxa and Chloropyron are pretty flexible about host, and some of these plants have been grown in the greenhouse using just the common sunflower, Helianthus annuus. We did attempt to grow Chloropyron tecopense to do a common garden study between the three population areas, but growing a desert Chloropyron turns out to be very, very challenging. And we got germination, some germination, but it was really hard to get them to grow into full-blown plants. Yeah.
So mostly grasses, and the grasses are pretty consistent across the three habitats?
Yeah. Across three habitats, you have Disticus spicata and Sporobolus aeroides, and those are likely the primary hosts. And then perhaps Juncus too, Juncus balticus, and maybe Juncus cooperi.
Thank you.
Do you know if it's an obligate hemiparasite or facultative?
I think they might be facultative.
Okay.
Yeah.
We have a question online. Natalie, go ahead.
Hear me okay?
Yeah.
Oops. Okay, perfect. Thank you, Naomi, for sharing that you're presenting this morning. I'm in a different time zone, so appreciate it. Please continue to do this. I guess my first question was on the data that you have on height and width. Like, when was that measured? I noticed that, I don't know if I got it wrong because I'm not really good at data analysis, but I remember the sample size was 13 from each site. And so I was just wondering, like, when you collected those data, was it June peak green, or was it just whenever it was convenient? Yeah, that's my first question.
Yeah, that's a really good question. The morphological measurements we collect, they're all from samples we collected, and we collected those samples all at peak, like maximum growth. So those would have been like before they went senescent. We collected them all around the same time period. So like in the month of it was like late August, early September. So they weren't still in an active growing phase, if that makes sense.
Gotcha. Yeah. Love the coloring on that graph. at the FST, I think. Yeah. Just a note. And what else did I have? Oh, I was just curious what gear you use to take these pictures. They're beautiful.
Oh, like this picture right here, I didn't take. I had actually a documentary film team out with me that took some pictures.
Wow,
It's professional,
that's awesome.
but I did take pictures of the plants
Oh my
with
God.
my iPhone and my DSLR. So there's a combination. Depends on the attribution
Nice.
of each picture.
Gotcha. So they did edit like these photos because I
They-
noticed that they're more like green, bluish than red. So yeah.
No, this picture right here that's still on the screen.
Mhm.
So this is taken by
Yeah.
the documentary film team, and these are seedlings. And this is I feel like true to color, where the lower leaves have a
Gotcha.
reddish tinge and they're kind of grayish.
Uh-huh.
So they're pretty gray plants. I would call them gray.
Cool. Okay, I remembered. My last question, sorry for taking up time. Is how long would you be out there for per trip? And I guess I like noticed that you sampled for like 6 months or so for the FST graph. Yeah, just wondering like how that works in terms of manpower and like timeline.
Yeah, we were out there a lot. I don't know how many times I've been out surveying many different months out of the year for these plants to kind of also, in addition to the data that's recorded on herbarium specimens, to get a sense of like timing and phenology and all of that.
Yeah.
So yeah, we did all the genetic sampling primarily in 2023 and 2024. And we did we did a lot of field work in like June, July, August, September.
Gotcha. Okay. Permission to post some photos on my socials. I won't be posting any of your data because there's no point really. But yeah, just to kind of get the word out and stuff.
Sure.
Thank you so much.
We have another question in the room up in Reno. Yeah, Blake.
Hey, you mentioned that, the relatedness between the Ash Meadows population and the Fish Lake Valley population might be explained by long distance dispersal. But you mentioned that you have maybe some other ideas. What are some of those other ideas?
Well, that's my primary working idea. And I think that it could be long distance dispersal. So when you think about perhaps what these wetlands might have been like during the last ice age, the last glacial maximum, and where was there hydrological connectivity and where was there not? So there was definitely potential paths for dispersal and gene exchange between Ash Meadows and Shoshone and Tecopa, because there were many more waterways that were connected. So there could have been right there, like they could have diverged, like as you had drying coming out of the ice age or something like that. But there wasn't a lot, I don't know, of any hydrological connectivity between Fish Lake Valley and Ash Meadows, like within the time frame of like these past glacial maximum. And so I don't know how else they would have, if perhaps Fish Lake Valley is derived from these others. And that's the thing, is so that my student Erica is going to be doing a biogeographical analysis to do some molecular dating to basically see the ages of the different lineages and to be able to better pare down the hypothesis of how they got to where they were and how old each population area is. And I don't want to speculate too much until we have some of those data.
Cool. Thank you.
You noted that the Ash Meadows had the highest highest genetic diversity, and so are some other plants as well. Is there some idea that might be the source?
Oh, that's a good question. So there's a question in the room if Ash Meadows because it has the highest genetic diversity, as does some of these other species, if that could actually be the place of origin for the species. And that's, I think, a really good hypothesis. It could be. And so that's something we'd like to investigate to see if ash meadows perhaps was the center and then they diverged south and north from there. I don't know. But that is a good working hypothesis. Yeah.
Do you recall which population had the Lycia blossom observed?
That was in Tecopa. So we found that in Tecopa Marsh. Yeah.
Could you also remind me which population had the enlarged corollas?
Fish Lake Valley.
Okay, thank you.
Yeah. I would love to see, what pollinators are in Fish Lake Valley.
I was just curious, maybe there was an association there, but you said that they're mostly self-pollinated, correct?
Yeah, according to the genetics, yeah.
Well, if there are no other questions, I'd like to thank you very much for sharing all this research and data. And it's fascinating to hear what goes into all the different areas of research you're doing to go in to try and sort out these different populations of Chloropyron. Really appreciate it. Thank you very much.
Thank you.
Thank you. Thank you.
Yes, we'll be watching out to see if they get listed. Thank you to everyone who attended. Our next meeting is the first Thursday of May, and then we take a break until September while people are out and about in the field. But we have one more meeting coming up in the spring. Thank you.